Instead of drawing a line between significant and not significant, GSEA ranks all 20,000 genes by a combined metric, typically the product of log 2-fold change and negative log 10 p-value, and asks, do the genes in a pathway cluster at the top or bottom of this ranking? The GSEA algorithm walks down the ranked gene list from most upregulated to most downregulated. At each position, if the gene belongs to the pathway of interest, the running sum steps up by a weighted increment. If the gene does not belong to the pathway, the running sum steps down by a smaller decrement. The enrichment score is the maximum deviation of this running sum from zero. A large positive enrichment score means pathway genes are concentrated at the top of the ranking, upregulated together. A large negative score means they cluster at the bottom, downregulated together. Is the enrichment score significant? Permute the gene labels, randomly assign genes to the pathway, and recompute the enrichment score. Repeat 1,000 times. The permutation distribution is the null expectation. The p-value is the fraction of permutations with enrichment scores as extreme as or more extreme than the observed. Normalize for pathway size. Larger pathways tend to have larger scores by chance, giving the normalized enrichment score NES. Apply feature correction across all pathways tested. The leading edge is the subset of pathway genes that drive the enrichment score, the genes that appear before the running sum reaches its maximum. These are the core contributors. If the inflammatory response pathway is enriched, the leading edge might contain 23 of the pathway's 200 genes. Those 23 are the specific inflammatory genes most changed in your experiment. Report them. They're your candidate targets, your follow-up list, your biological story. GSEApy is the Python implementation used in your spatial transcriptomics pipeline. It supports fgsea from a pre-sorted gene list, the Enrichment for quick ORA against hundreds of gene set libraries, and ssGSEA for single sample scoring. fgsea uses a fast approximate algorithm that's orders of magnitude faster than the original permutation approach. Your pipeline uses GSEApy with a 3-tier target scoring system, Tier 1 for druggable targets with known compounds, Tier 2 for novel targets without existing drugs, and Tier 3 for known pathway members useful for validation. Single sample GSEApy scores each sample or cell for pathway activity, producing a continuous score rather than a binary enrichment call. In single cell analysis, compute pathway scores per cell and overlay them on the UMAP embedding. You can see which clusters activate the interferon response, which activate the cell cycle, which are metabolically active. AUCell is an alternative that uses the area under the recovery curve, robust to dropout and scalable to hundreds of thousands of cells. Pathway activity scoring turns cluster identity into functional characterization. AURA asks, are these genes enriched in a pathway? GSEA asks, does this pathway cluster at the top of the ranking? Leading edge identifies the core drivers. ssGSEA scores individual cells. The library catalog turns a gene list into a biological story, from 500 uninterpretable genes to 3 enriched pathways with 23 actionable targets.